So once again, a very warm welcome to all the audience present in this lecture on the platform of Center for Human Resource Development, CHRD, Dr. Sarupalli Radhakrishnan, Rajasthan Ayurvedic University. This center is established by Honorable Vice Chancellor, sir. It is his dream vision, and it has been established in terms to the orientation of the faculty, of the students across nation, and regular programs we organize on this platform regarding the orientation programs and various lectures on various issues, various vulnerable issues. And in this context, this center is organizing another lecture series on technology-based evidences for Ayurvedic solutions in precision health in collaboration with JCKC and IIT Jodhpur. This is the second lecture. Earlier we had a lecture on in January month, and we hope that due to some problems that the students were having their exams, and in such period we couldn't manage the lecture in the lecture in the previous month. A little bit of delay has been there, but now onwards we will keep on moving regularly in this lecture series. I'm very much thankful to our Honorable Vice Chancellor, Sir, Professor Dr. Abhimanyu Kumarji, for his constant guiding and motivation regarding moving ahead in the surge of finding new solutions, finding new knowledge, and presenting this before the Ayurvedic fraternity. I also acknowledge my deep gratitude to Professor Shantanu Chaudhary, sir, Honorable Director, sir, IIT Jodhpur, for motivating us to move ahead in this lecture series. In our original lecture, in our inaugural lecture, both the dignitaries blessed us with their motivative speeches. And now today we are moving ahead with the second lecture, that is on micro-nano sensors for sensory perception-based Ayurvedic diagnosis. This is a very new idea, and university in collaboration with IIT has started a project Ayurtech, where we are working in the field of taking some integrative approaches in developing some tools, in developing some such type of methods that we can incorporate the technology intervention in the field of Ayurveda. And various steps has been taken, and this Ayurtech project has been sanctioned by the Ministry of Ayush, and a center of excellence has already been established in IIT Jodhpur. And the principal investigator in this project is Professor Mitali Mukherjee. Now today, as it is a great day, it is World Health Day today, and luckily we are organizing this lecture on this day, and we think an integrative and precision and integrative approach in the field of precision health will be taken up. And in this regard, today Professor Ajay Agarwal ji will be delivering his distinguished lecture. Professor, I would like to, before I invite Professor Ajay Agarwal ji, I would like to share a brief bio sketch of Professor Ajay Agarwal ji. He is a very distinguished personality. He is professor at IIT Jodhpur, associated with the Department of Electrical Engineering, School of Artificial Intelligence and Data Sciences, and Division of Interdisciplinary Research Program IDRP of IIT Jodhpur. His other responsibilities include Senior Chairman, Engineering Science Undergraduate Committee, President, Institutions Innovation Council, Member, Research Committee, School of AI and Data Science, IIT Jodhpur. He is co-principal investigator for Center of Excellence in Biotech for Integrative Precision Health and Medicine. Earlier, he worked at CSIR, Central Electronics Engineer Research Institute, CD, Pilani, as Senior Principal Scientist and Coordinator Smart Sensors Area, and Head Nanobio Sensors and MEMS and Microsystem Groups. He is Associate Dean Engineering and Sciences at Academy of Scientific and Innovative Research, ACSIR, New Delhi. He also served at Institute of Microelectronics, Singapore, as member of technical staff for over 9 years, and SCL Chandigarh and Usha India Faridabad for over 6 years. He is involved in the development of nanotechnologies, MEMS, microfluidics, and microsensors for environmental monitoring, healthcare, and industry 4.0 applications. His engagement with semiconductor industries, research institutes, and academics is for over 3 decades. He has more than 290 research publications in peer-reviewed journals or international conferences, 100 invited plenary keynote talks, and over 35 patents granted or filed. He has supervised 15 PhD students and is guiding 11 PhD students currently. He is senior member of IEEE USA since 2006 and vice chairperson IEEE Rajasthan subsection 2021. Life fellow and chairman Pilani of IETE India and Semiconductor Society of India, life fellow of MSI India and the Institution of Engineers India, and member IAM International Association of Advanced Materials Sweden and MRS Singapore till 2000, and associate editor Healthcare Technology Letters, etc. He has been bestowed various awards, including 2021 IETE Bapu Sitharam Award, Outstanding Engineering Services to Society Award 2021, the Institute of Engineers India, Rajasthan State Center, Jaipur. 2020 CSIR Technology Award Innovation. 2013 DST UK ERI Thematic Partnership Awards India. 2009-2008 National Technology Award Singapore. 2009 Excellence Award IME Singapore. Collaboration Development Award British High Commission Singapore 2005 and 2006. And Super Kaizen four times and Best Kaizen 7 times at Usha India Limited, etc. His biography is included in the Who's Who in the World, which profiles the most accomplished men and women in the world since 2010 and Who is Who's in Asia since 2012. So with this, we would be able to know the vast experience and his distinguished personality. And now I request Professor Dr. Ajay Agarwal ji to please start with the today's deliberations. Professor Ajay Agarwal.
Thank you, Dr. Rakesh, for a nice introduction about the university and about myself also. Thank you very much for the same. I would like to, I'm starting my presentation. Is it visible? Can someone say that is it visible? Am I audible?
It is visible, but something, a patch is coming over, a black patch is coming over there, which is making some disturbance. Yeah, where you are putting your mouse right now.
So here is a picture here, so maybe I have to move it to some other position. So
yes, okay.
Okay. Yes. So thank you very much for giving me this opportunity to talk about my work and how it can be related to Ayurvedic diagnosis. As you mentioned, this is a very good initiative about Ayurvedic Center of Excellence, which has been established in IIT Jodhpur, and Ayurveda University at Jodhpur is one of our main partners in this activity. I would like to thank all the organizers, the Vice Chancellor of the University, Professor Abhimanyu. I also want to thank Professor Shantanu Chaudhary for giving us this platform to work together. So the topic what I have chosen today is really interdisciplinary topic, and I assure you that I'll not uh, bore all my participants here or audience here with too much of electronics here or too much of technology. We'll try to understand and see how these different technologies are being used or can be used for different diagnostic applications. Uh, Pardon me, because I am not an expert of Ayurveda. I started learning about this 6 months back only, but I've been in diagnostics and biosensors development for over 15 years, more than 15 years. So I am using that knowledge for this field of science also, medical science, which is Ayurveda or Ayurvedic diagnostics. So our Jodhpur colleagues all know that this is a very good campus, which is next to almost 2 kilometers from Ayurveda University in Jodhpur. So this is a front building, administrative building, and if you go along the path which is visible here, almost a kilometer inside, you will find all different kind of departments, and this is a real Jodhpur stone buildings and fantastic campus we have. And what you see here, it is not that green, but today it is really a green campus, and it's not that brown what is visible from this picture. And left hand side, what you can see, there are so many different departments, which gives us opportunity to work interdisciplinary work together, understand each other's science and engineering aspects, and complement each other in many developments. Uh, so we have many things here. We have bioscience, bioengineering department also, which is very important for any diagnostic application. Their knowledge is important. But when we are working for, say, Ayurveda as a our target application, so the presence of Ayurveda University next door to IIT is a very welcome sign, and that encourages us to work together. And we are almost, as many of the doctors in the meeting today understand, that we are almost meeting every 2 weeks or so and discussing on different problems and trying to look for the ways to put uh, meaningful solutions for the problems being faced today. So this is a brief agenda. So you can see that I'll start with some understanding of Ayurveda and why this technologies can help in that Ayurveda, and then various aspects of biosensors and technology aspects I'll go through. So as I understand, Ayurveda is natural healthcare system. So this is a comprehensive system of natural healthcare that originated in India and Vedic times. So this is our own system, and we should not neglect, and what we feel we have to strengthen is so that the world recognizes it to the best possible. So in Ayurveda, when we go through, it emphasizes on prevention of disease and maintenance of health. So these are two basic things where emphasis is there when we go for the Ayurveda. Original knowledge based on Ayurveda is based on evidence. So it is all based on the evidence. So evidence is one most important aspect. And evidences are described in context of praman or pramana. When we say in Hindi, we can say praman. So based on praman, and there are 4 categories of praman, what I understand. And these pramans, for the understanding of other people, it is a right perception. So it is the right perception or the praman of condition, and based on that, diagnosis is done. So what are the pramans? And these are the 4 categories briefly. The first category, as I understand, is prataksh. Prataksh. Pratyaksha means evidence generated from the actual and direct observations. And these are measurable, and when they are measurable, these are measurable from direct feeling and understanding through five senses. So five senses are very important, and what these senses are feeling or understanding, that is most important aspect. And all these evidences are measurable. So this is something pratyaksh. Whatever is visible to you is a pratyaksh praman. Next is anuman. So the second category, which is anuman, is it involves the analysis of the observed and measured data. So measurement has happened in the first aspect, and that need to be analyzed further. And how this analysis is done based on the logics and causality. Third one is apta, and which comes from the experimental wisdom and unbiased opinion of experts. And the fourth here is yukti, which is a tactical analysis. So if we see, these are the four categories of praman based, or the evidences based on which the diagnosis is done. So data is collected from the first one, and here the senses are playing the important part. So what we sense, that is the data, and that need to be analyzed further for the diagnosis purpose. So what are the five natural senses with us? This is a God gift to us. So we have five senses, and similarly there are five kind of stimulus, and these stimulus are mechanical, chemical, electrical, light, and temperature. These are the only five senses or stimulus. And sensory perception begins, they start sensing when something stimulates our sense organ. So something is stimulating, and for example, light stimulates our eyes. So if there is a light falling and the reflection comes from there, it stimulates our eyes. So sensory organ is stimulated by this stimulus. Warmth or the heat. So if the cup is hot or cold, that stimulates the touch feeling, touch senses. Similarly, a combination of stimulus is also possible, like chemical, mechanical, electrical, and temperature. These stimulus can generate a pain. So the pain is because of these combination of stimulus. So there could be many combinations or single stimulus which can trigger some of the senses, our sensory organs. So chemical stimulus, if you see, can be perceived by both sense of smell and taste. So our nose or tongue, they can give us a reflection of chemical stimulus. So these are the stimulus coming up, and how our senses are reacting to these stimulus, it is important. And why there is a need of sensory system, when I am saying that there is a need of some sensors, because these stimulus, how our senses are responding to these stimulus, that is a basis for the diagnosis, that how our body is responding to various stimulus. And if our different senses are not responding to different stimulus, that means there is a problem, and that is a kind of a disease condition, and that need to be treated. But if our senses are not working properly, then what to do? So human sensors perception tends to become weaker with aging. So this is one reason that with time our senses may not work perfectly as they are supposed to do. Individual perception of various stimuli may also vary. So some person can smell a particular smell very prominently, while the other person may not feel a smell at all in that particular environment. So that doesn't mean that smell is not existing there. So that's an individual perception variation is there, and that may lead to inconsistency in the treatment also. That's my way of thinking. So there is a need for alternative sensory system for accurate diagnosis of different stimulus. The system should be, and this system should be accurate and should provide standard outputs. There should not be a huge variation in the output so that it becomes difficult for our doctors or practitioners to take a decision based on that. Fine. So that is a need, why there is a need of some sensory systems, alternative sensory systems to help further diagnosis. That is what I'm trying to develop here. Now, when we are making these sensory systems, alternative systems, can nanotechnologies be used for the healthcare? Yes, because these nanotechnologies or nanomaterials are important for sensors because they provide very sensitive sensors. So they improve the sensitivity of the sensor. We'll see that part also with some examples later on, how we are able to improve the sensitivity of the sensors when we are using these nanotechnologies. New signal transduction technologies in biosensors are possible. When we say signal transduction means there are alternative ways to detect different chemicals or different signals, the stimuli. So different stimuli are detected in an alternative ways. I'll give some example of these also. Uh, and when we are making these sensors or the system smaller because of the nano sensors, the peripheral electronics or the other instrumentation also reduces in dimension, and that helps to put these sensors or probes into the body also very easily. So for in vivo analysis, because of these small sensors, now it is possible to put them inside the body also. Like nano capsules you might have heard of. They have some cameras, they go inside the body, they do a continuous imaging, and then they come out from our gut. So that is possible. So they give a very accurate testing, and other things can also be done, and which is being explored today using these nano sensors. And when all this sensor and peripheral electronics is reducing in the size, the overall instrumentation is reducing in size and it becomes portable. So when we are going for some testing in the labs, many times we see a big instruments there. Today there is a need of portable systems, so they are easy to carry from one place to another, and they can be taken to the point of care also. Right? So what kind of opportunities we have? So these are general solutions. What I'm trying to see are the opportunities, and based on some of the examples and what kind of development going on in the diagnostics in the healthcare sector, mainly what I will be touching upon, they can be or they are being explored in our team for Ayurveda also. So I'll touch upon wherever we are attempting some of these diagnostics for healthcare in Ayurveda. So when I'm saying about the nanosensors, healthcare diagnostics, in healthcare diagnostics, these are the two major areas where we have opportunity today. One is early diagnosis and point of care diagnosis. Early diagnosis means, if you see the early diagnosis, these are some of the normal use cases. Say heart attack happens. Somebody is having a chest pain. We want to rule out all possibilities of this pain. See, whenever there is a pain, the first assumption is maybe because of some gastric problem, he might be having a pain or some muscular thing might have happened, but it might be an early stage of a heart attack. Fine. So if we can understand that heart pain at the early stage and can rule out if it is because of some heart problem or other problem, the life can be saved. Similarly, for cancer diagnostic, early diagnosis is a major requirement. Most of the time, what happens in the cancer case also, the patient reaches a doctor at a very later stage, maybe a third stage. And first, second stage, when he has some problem, some fever, some inflammation at some portion, he may not even consider it as a chance of cancer. So normal medication is taken or it is being neglected also. So there is a need for early stage. COVID, as we have seen, that has affected all of us worldwide in last 2 years. So always there is a need for early diagnosis, and early diagnosis means see, the test as we started with, it was taking 24 hours or 48 hours initially, and later on some different solutions which have come. So instantaneously or within few minutes we were able to detect it, and we were um, trying to solve some of the problems related to isolation of those people and other things. Similarly, there are many diseases like Alzheimer, dementia, diabetes. They need early diagnosis. So in case of Alzheimer or dementia, if a person is understood to getting affected by these conditions at a later stage, probably he may adopt to a lifestyle which will not affect his life too much from his normal living. Similarly, diabetes, if detected early, because now it is affecting we people at 30s only. So people are becoming diabetic at the 30s. Diabetic people are there in the children age also, but in 30s or early 30s, we are seeing a large number of cohort going to be diabetic. So if you can detect early, they can probably change their lifestyle, the proper medication can be taken, and sometimes without medication, and what I feel like the yoga and the lifestyle which is prescribed by the Ayurvedic way of treatment. So without treatment, actual medicines, the people can have a healthy lifestyle. So early diagnostics is very, very important for many, many conditions. And if we are having early diagnostics, definitely chances for lethal diseases like cancer, these patients can survive longer, or the chances of getting complete recovery is increased. And also the time for the cure can be reduced if they are treated from the very early conditions. And when we are doing it quickly, it is reducing the cost or the burden of the treatment. So these are some of the major things we should consider always. If you diagnose early, it always has an advantage for the extending the lifestyle or extending the life of the person. And what are the challenges here? Why we are not able to diagnose early? So for early diagnosis, each disease has some markers in our body. These are some molecules in our body which are in circulation, but their concentrations are so low, they are difficult to detect. Or if we want to detect also, their procedures are difficult, complicated, or very costly also. So we are not detecting just for sake of understanding if we have any deadly disease. We are not able to put thousands of rupees, and when the markers are in very low concentration. So markers are there. I have worked on some of these markers in very low concentration for some of these conditions. I'll show you some of the results later. But there could be many, many markers for many lifestyle diseases or other diseases, deadly diseases. And if they are in low concentration, using these sensors, probably we can diagnose or detect them at very early stage, and uh, we can prevent. And some of the existing techniques are taking a lot of time also. So this time is also very crucial. For example, if a patient is in ICU, and if that patient is suffering from, say, fungal infection or bacterial infection or some viral infection, it takes some time to detect, to analyze in the pathological lab. And if you can do it in, say, minutes, you can give an accurate medicine to that patient and you can save the life. You can avoid unwanted medications also. So today, if the doctor is not able to diagnose immediately for such critical patients, they have to prescribe some medicines or multiple medicines on some gases. Anuman, on some gases they have to, but they don't have an exact diagnosis of the. And there is a negligence also at early stage, I'll say, because of these factors, high cost and other thing, or we are busy, we neglect some of these signatures at the early stage, and that becomes a difficult situation at the later stage. So these are some of the challenges what I found are stopping us from the early diagnostics. Other thing, what point of care? So we want the care or the test to be done where the patient is. It is not that patient is going to the hospital or a clinical settings or need to be taken to some other dispensary for the test. So many of these tests need to be done there only where the patient is. And this is very, very helpful for many conditions. And I am quoting some of these kits which are available today, and they are changing the life, our uh, the way we are doing the things today. So glucose testing, as we all use today, so every household or every next household, there is someone who is suffering from blood glucose problem. So they need to do a testing at some intervals, maybe daily or twice a day or once a week, whatever is the condition based on. But now today they are not supposed to go to a clinic for that test. So they can do these tests very easily, and similarly there are many other tests. But when we say um, point of care, there are 5 different conditions of point of care. These are the 5 conditions. The first one is home. So what I mentioned about the glucose testing, it is at home. This is called self-estimation. So we know that we are estimating, we are not taking medicine based on these tests. No medication is supposed to be taken based on self-test at home, but they are important for self-assessment for the control purpose. So you have a gadget, you keep checking your whatever condition is possible to be checked at home, like glucose condition. So if you are in, say, 100, 120, you are happy. And if it goes to 150, 170, you may have to call a doctor and he will say that, okay, fine, you take that medicine, or you have to do something change in your um, dietary habit or other things. So that's something like self-assessment. And there are many examples. I don't want to quote it, but there are many of these things, and they are very, very helpful for routine testing. Even pregnant ladies are doing some of these tests at home today, and these are helpful for monitoring various vital functions, and that helps them to carry on their say routine life, apart from going to a doctor very routinely every week or every 2 weeks, something like that. Next scenario is a community-based point of care devices. So this is something like we go to a school, go to a village, we go to a community or a cohort somewhere, and we do analyze people there for a particular condition, and screen out people who are suspected for a particular condition where we are looking for. So we screen these people. Again, we are not providing medication to them. We screen them. All susceptible people for that conditions are then referred to a doctor, and they do a thorough checkup. And whoever is really positive in that cohort, the selected people, will be given medication. Otherwise, all the people many times like tests in the schools. So thousands of students may not be going to the clinic for the routine checkup, but a practitioner can go. So these are not doctors, these are the health workers only. They can go to a school, can do some test, quick test. These are screening tests mainly, and they can diagnose some of these who are having a particular condition or likely to have that condition. The third condition here is on the doctor's table. So doctors do some test, like they have a stethoscope, they have BP meter, they have temperatures. So similarly today, and I have seen this condition in Singapore and other countries also, doctors have a lot of kits on their table. So they do some blood test, they also do some urine test on the spot, and based on these tests, they prescribe the medicine. So they are not referring patient to some clinic for some tests to be done. They do some basic tests on the site. So they take 10 to 15 minutes per patient. They do all these tests and then they prescribe the medication. The fourth case is quite common in India. So peripheral labs. So doctors need to prescribe the patient to go for some tests. They go for diagnostics there. But in diagnostics also, now we are looking for quick answers. All the tests to be done and the report should be given as soon as possible. So these tests which are done in the peripheral labs need to be done using the techniques which are fast enough, and that's how these are finding these new tools, new diagnostic tools are finding a big space in peripheral labs also. Case is in the ICUs. So ICUs, the patients are critically ill. They cannot be referred or they cannot be taken to the laboratories which are in the hospital, in fact, but they cannot be transferred to the lab. And whenever the doctor goes on the round, he wants some of the tests to be done there only on the spot so that he can check the condition and prescribe the medicine or alter the medicine as required by the ICU patient. So inpatients typically in ICUs, they also find lot of applications of point of care devices. So these are the 5 cases, and when we are having these 5 different cases, their structure, the system requirements may change. So there are all these are systems. So what these systems are made of, we in general say biosensor, but this is a system. This is quite busy slide. Please don't worry. Let's start with the leftmost vertical here. These are the samples we have. So for human samples, we see blood is there, or urine is there, or saliva is there, or cell. So these are our samples. They need to be analyzed for color, smell, which are more prominent for our Ayurveda testing. For modern medicines, color is important. They do some analysis of the color, but smell, I think they do not bother much. They look for molecules inside this one. But for Ayurveda, color is important. Smell is also important for various body fluids. Body smell is also important. And yes, cells or the tissues, they are being analyzed for many things. So these are important basic samples for human and diagnostic purpose. But other samples are similar, so I have put them also here, the food samples and environmental samples. So they are also similar, so I have put there. They are also being analyzed using the same type of systems. Now, how they are being analyzed? Here, if you see this one, the electrical interface or the sensors, these are the transducers. Transducers means, see, most of these molecules are charged particle. Say if I am looking for some charge particle based sensing, means if I can bring that molecule on an electrical sensor like this, and if it sits here, if because of that charge, there is a change in the electrical property of this sensor, I should be able to sense it. So that is what we try to do. We make these gadgets, these sensors, which are electronic sensors, and these have nano structures. And being nano structures, they are really very, very sensitive. Even some of these sensors are sensitive to single molecule. So if single molecule of any kind comes on some of these sensors and it do some changes, and we are able to detect that those changes, you can diagnose these molecules. So how to specifically bind these molecules on the surface? These are the receptors. So that's why our chemist or biochemist help us to bind these target molecules. So we understand what molecules we want to detect on our sensor. So they design these molecules for us. We put these molecules or bind these molecules on the surface. Now, when I am decorating this, say this is a nanowire with a molecule here, which is selective for a say target molecule in my sample, only that molecule will bind. All other molecules will not be able to bind on the surface, and they can be washed off. So very selectively we can bind the target molecules on nanosensors using bioreceptors. Now I am saying few molecules or single molecule need to be detected. So when a small molecule number or single molecule is coming, the change because of those molecule is also very, very small. So there is a technique called signal amplification. This is all electronics part here. So we make some circuits so that we can amplify these signatures, and they are then readable. And those signatures need to be processed also. That is through a processor. Processing, why processing? Processing is something like what was the signature this sensor giving before. When I bring this uh, bioreceptor on this and molecule comes, there is some change. So I want some processing. If I have multiple, say 3 wires here, they are having a 3 different bioreceptors, and 3 molecules can be detected together. So there are 3 signals coming out. So I need some more processing here. And similarly, we can do hundreds of such processing together. I'll show you some pictures of the devices which I made in my team, and we can have hundreds of such sensors on a single chip, and all can be processed together. And when we are doing processing, is it a meaningful or not? What we are hearing today, artificial intelligence. We give some intelligence to these processors so that whatever is coming out is more meaningful. Because there are a lot of interference also. We are looking for few molecules, and there are hundreds of unwanted molecules. They may also give some kind of noise. So we have to remove all noise signatures from the system. So for that purpose, AI plays an important role. So combination of all these knowledges together makes a system, and that helps in detection. I'll not go in the detail of this one. When I said the charge particle, it is called potentiometric transducer. Means the change of the sensor is dependent on the charge. In some cases, there is some reaction happening. So some current is produced. So such sensors are called amperometric sensors or transducer. Transducer is something which converts one kind of energy into another. So because of that molecule, some reaction happened on my chip and some current produced, and the current in the system changes. That is ampere. There is some colors. Colors we all understand that we put, there are a lot of available in the market. You put a sample, it flows, And if a red line comes, if a blue line comes, so there is an optical signature there. So color comes, and based on the color, we can distinguish. Similarly, acoustic transducers are there. If a molecule hits all the nano surfaces or nano, or in this particular case, piezoelectric crystals we put in the sensor, their oscillating frequency changes because of very small quantity of the sample. So these are four different principles mainly on which all transducers work. There's various variations here. I'm not going the detail. Uh, just have a look at some of these nano sensors or the nano wires. These are all pictures from my lab, which I was doing in Singapore. These are not in India. We were doing in Singapore that time. So these are the nano wires. So multiple wires you can see, a mesh of wires or vertical wires. And these are very, very long wires, hundreds of micron, even millimeter long, and each one is having an electrical contact separately. So we were making hundreds of such wires on a single chip. I'll show you some more pictures where we can do these sensing applications. Fine. So now, as I mentioned, this is a cartoon of say optical sensor. So this is my chip. So I have already placed some chemicals here. When I put a sample on it, there must be some reaction happening. And I put it into a reader. This is my reader. And if the color change happens, there are electronics here which can detect that color and can give you a signal. So is it a positive signature or a negative signature? And if you want, it can be transmitted to your mobile phone, to your doctor's mobile phone. So depending on what kind of feature you want, we can make these type of systems today. And these are small handheld type of systems. Similarly, if you have an electrical change, so these are some electric here, you put here, and when you put it inside, there is electrical change happening, and similarly the signature can be sent. These are some of the platform devices we have been working in my team, and I'll just show you some of these pictures quickly. So all charge-based sensing, I mentioned 90% of the molecules have charges. So if you are able to bind them specifically through a bioreceptor, these are SFET devices or extended gate devices or silicon nanowire devices. These are also CNT based devices, or these are some other versions of electronic devices. But these are all sensitive for, they have a different sensitivity also. I can tell you, and we can tailor the sensitivity also. So they are sensitive to charges basically. So any charged molecule, if you can bind specifically, they can be detected using these sensors. These are some other sensors. So this is a particle lab on a chip where the particles can be separated and the counted also automatically. How it is happening? So we designed this one. This is I'll show you some more details of this one. One more slide is there on this one. So this is for the blood particle separation. So when you flow blood from here, WBC arrest in this microfluidic channel here, and RBC are arrested in this portion, and plasma comes here. So how this separation happens? Because of the filters on the side. So blood is flowing in a channel, and when it is flowing, there is a separation also happening. And by seeing how much channel is filled, we can estimate the count of these RBC and WBC. Right? This is a small chip, again, very small chip. Viscosity of the fluids can be measured just by a drop. You have to drop a liquid here, it flows through, and depend when it is flowing through. So there are different designs as you are saying. When it is flowing, we can estimate. There is electronics also, which I am not showing here. From the flow, because more viscous liquid will flow slowly. If it is less viscous, it will flow faster. And from that flow rate, we are able to estimate the viscosity of the fluid immediately on the spot. And these are some other devices. So these are some micro electrodes, and this is having a nano channel here. So you put some chemicals here. You can control the flow of these chemicals using the electrode, the third electrode, and you can stop also, and you can do a very controlled experiment in these type of devices. The other devices which are also useful for diagnostic are, these are the gas sensor, the smell. Different smells can be done. I'll show a couple of slides on this one. This we have been doing. This is again another device which has the nano gaps, and this device is also very effective for detecting single molecule, and we are getting a very good signature of single molecule using this type of chip. So that is the ultimate capability of this type of devices. And this is that ultrasonic transducer. So these are on chip. So all these are chip based. Uh, so if you mobilize, immobilize your molecules, the frequency changes, and that can be detected. And these are pressure sensors. So pressure sensors chip are also important, and they are being used for many applications. Let me take a couple of examples. Um, what we have, gas sensors. So gas sensor, so environmental monitoring is important. So many gases are being done, and these gases are important to be detected. But they are also important for our breath analysis. And as you know, for these conditions, our breath has some of these gas coming up. So if uh, as I have been discussing with many doctors also, different patients, different people come with a different breath. It is not only because of some decay or in the mouth or in the tooth decay, but these are signatures of our metabolism happening within the body. And these gases can, if you can detect them in very low condition, they are signatures for many diseases, and that is how this could be very handy tool for screening. So this is what we make this type of chip. This is about 2 mm. We bond it. This is a device. This is a system. Then uh, they work on the principle like they have some resistance, and when a gas comes, gas is absorbed on the surface, and depending on it's a reducing gas or oxidizing gas, the resistance can increase or decrease. So in this case, this is ammonia. So you can see 17, 8.54. So we are reducing the concentration. You can see the change also is happening. So you switch it off, it comes back. So this is a type of, and these all corrections can be done, all those things. So, and these material here, the sensing material, there are different kind of sensing materials indicated here, and they respond to different gases at different temperatures also. So this kind of sensitivity improves at a particular temperature. So like this first ammonia on tin oxide based sensing film, the maximum response comes at 220. So this is the sensitivity is maximum. So by seeing the patterns of different gases for different sensing films, we can distinguish multiple gases in an environment also, and we have done that part also. I am not going the detail. So these are some other calibration curves from low concentration to higher concentration, so 10 ppm to 50 ppm, and you can see how the response is increasing, and this can be used for various detection, and in fact various VOCs or inorganic gases, these all can be detected using this. And these are something like a mobile phone type of systems, or these are as small as like our watch, wristwatch, and they can detect various gases, and their output can go to your mobile phone also. So this kind of application we have demonstrated. Nanowire I have already mentioned. Advantage here is you can have hundreds of these wires on a single chip. The different colors means they have a receptor for different molecules. In the sample when flown through it, which might have a different molecules. So when they bind in different target sites, so blue will bind to blue only, red will bind to red only. and then you watch it, and you can measure the current before and after, and you can find out that how much change has happened or which wire has changed, and that way you can detect specifically a multiplex molecule detection in a single sample. So these pictures I have shown. So there are a lot of publications we have done already. So these are indicated here. So this is on how to make such wires, and these are hundreds of wires here. This is a particular chip I designed for a company in Australia. So there are some 36 clusters. Clusters means each group is having 5 sensors. So there are 36. So we wanted to detect 36 species in a single go. So 36 into 5, so that many sensors were there. To measure so many sensors together, this is actual picture of the chip. You have a pads here, and you can flow your liquid continuously here. And this system was made, or which was a tabletop system, which has all electronic system arranged continuously. And this is by spotting, you can add the receptors also specifically. So this is how it was designed. So these are the actual pictures. So this was a system developed for this type of application. And some quick examples I'll show that why I am putting a receptor. So if my charge is floating, so we started with pH sensing, that is H ion concentration. So if this is a cross section of that sensor. So wire is there, and there is some insulator on the top, otherwise it will just flow through it. We don't want the charge to flow through that wire. So there is an insulator layer, a thin insulator layer. So, but this charge need to bind to the surface. If it is floating, you can see the effect is less. The pink one is the floating charge. Same concentration, but floating charge. So like pH2, you can see a pH2, here it is. So the floating charge is giving a change of something like 10%, but if it binds, it gives 30% change. So that is how it is important to bind the charge. And this work was further demonstrated in another paper when we are having this DNA. So we made 7 different probes, and we made them to bind from the first base pair onwards, second base pair onwards. So we are one base pair, that is 2 nanometer, we are moving from the surface. So when we are moving systematically 2 nanometers away, you can see the output was decreasing systematically. So that's why the binding of this charge to the sensor is important, and these binding molecules also need to be as small as possible for output to be the maximum possible. This is another one of the most cited papers of mine, where we first time see the change in the mutation in the DNA, a single mutation. So this output here, the conductance is because of the 1-to-1 matching of target DNA and the probe. And when there is a one base pair mismatch, you can see from black it comes to red. And if you have two base pair mismatch, it further goes down. So long back in 2007, we demonstrated, and this has more than 500 citations today. So this type of small changes are possible. Similarly, I mentioned about different molecules. So this is related to troponin T. So again, this is all published work. This can be referred. What it is showing, 1 femtogram per milliliter. This is the red one. So we could detect so low femtogram condition of troponin T, which is responsible for cardiac condition. So this is how we did the modification and attachment, but we could continuously detect these different concentrations in very, very low. Further, I'm not taking many examples here, but let me show that we did it for cell culture also on these type of sensors. And this is a small video which is showing these are, if you can precisely, these are the live cells, they are beating. And if you give different drugs, they respond immediately, and underneath sensors can give you these type of signatures. So we have done a lot of drug analysis or responses to the cells. So these are the live cells, and you give drugs, and you can monitor real time how the cell wall opens and cell wall respond to these drugs. This work of again published by one of my PhD students in Singapore. This is another cell and a response to another. These are the cardiac cell. That was the muscular cell, and then we did from the live heart of a mice. So we did a surgery and took out a mice heart, and from the surface we recorded this is the ECG like signal, and this is the actual ECG recorded of the same heart. So they are almost similar. Purpose is from the surface the small charges can be detected and using this type of sensors. And this is one example of lab system on a chip. So most of this testing requires a lot of blood processing. I'll finish in another 5 minutes. Uh, so here is a raw blood pushed into it. The processing happens here, and only the plasma goes where the sensitivity is tested, and this is for this cardiac detection. And the challenge was given, can we do the entire processing on a single chip? Something like this we made in the lab. This is the actual picture, and do this testing in 40-45 minutes. So we could do it, and that's how we did it, and this is a patent here. This is the other sensor what I mentioned. So see, challenge is these are very, very sensitive and they have nano gaps. What we are doing here is we are doing a Raman spectroscopy. Raman spectroscopy is a good technique to detect different functional groups or molecules. But when their concentration goes low, Raman cannot give a signature because Raman signature is very, very weak. So what we have done, we have made these nano gaps and large number of nano gaps in an array format. And when we put our sample where the molecular concentration is very weak, one of the molecule going in any of these nano gaps is good enough. And when I shine my laser on this one for Raman spectroscopy, there is an enhancement in these gaps by 1 million times to 1 billion times. So depending on how well it is placed, the amplification happens and how well I am making these gaps. So this amplification happens from 1 million to 1 billion times. So that means one molecule is equal to 1 million molecules. Now my signature is good enough to be captured by Raman spectroscopy. So this work is also being done in IIT at this moment by some of my students. These are some of the work we have done earlier, and this is for cancer. The breast cancer we have done in consultation with Tata Memorial Hospital, Mumbai. So a challenge was there are 3 markers there and all need to be detected on the bedside when the doctor is doing the surgery. So that kind of challenge. So we wanted to do this testing and we could do some initial good tests for that purpose. This is a very recent work for the COVID detection using our saliva. So this is the actual spectra of these 9 patients, and they are almost similar, and this is the control, and which is very different. So we have done before doing this work, we did a thorough study of some of the proteins on this COVID coronavirus, and we did AI also to understand our sensor response to those peaks which are corresponding to, like these are some of the peaks which are corresponding to this coronavirus. And when we did for patients 100% it matched, and these are all control and none of them is similar to this, and that is how we can detect from the saliva, and this was an instant analysis. This work was also published in some other. So similar techniques used for other thing. This is what I mentioned about the micro viscometer. So again, sample volume is very small. You just need a bigger drop of it, and wide range of viscosities can be handled. And we have designed various, and with the proper electronics, which I am not showing here, we have done, and we can record it, and instantaneously we can tell the viscosity, which is important for many diagnostics for the medical applications. So we design it in that way only. Otherwise, viscosity in the industry is measured using other techniques. These are some micro electrodes I showed. So these are micron gaps, and there are different versions and various designs, and they have been used for current-based analysis of many samples. This chip I already mentioned. Probably I can just show one small video here. So there is a separation happening. So the fluid is flowing, and you can see the smaller particles. The RBC is collecting in the outer one. The first one is arresting the WBC, the bigger particles, and the plasma comes to this one. So this is how we can instantaneously do a quick separation, and depending on how much it is filled, we can estimate the RBC count also, which is related to hemoglobin also of the patient. So, and these are all transparent. So under the microscope, there is no need of microscope. Under the normal mobile phone, you can see that how much it is filled, and then it is giving you an estimate of RBC and WBC. That's all from my side. I conclude there are different techniques, different sensors are there, and they need a different interdisciplinary approach. Different sciences need to come together, and we have to understand the problem also very well, that how we can provide a better solution for the problem of our interest. And while we are making or choosing a platform, because I have shown a few platform devices here, we have to clearly see what are the markers and which property we want to use for the detection purpose, and accordingly platform need to, and what concentration we are interested. Always high sensitivity device is not required. If the concentration is higher, we can go for a medium sensitive device also, sensitivity device. Thank you very much. And this is a link where all my publications are available. So this is a Google Scholar link, and you can have a complete citation of my research work or my team's research work. Thank you, and I acknowledge the contribution of my colleagues and students who have worked with me at IIT Jodhpur. They are working already, and earlier with me at Siri Pilani and IME Singapore. Thank you very much. I'll be happy to take any questions. Yeah.
Thanks a lot, Professor Agarwal ji. This has been a very something special from our point of view as far as we fraternity of Ayurveda is concerned. Specifically, we are concerned with our whatever we are concerned with our studies. And technology intervention from your point of view, because you are the technology experts, and how we can manage and how we can correlate the technology, in particularly in concern with the diagnosis issues. This is naturally a very interesting topic. As you mentioned, various type of technologies have been developed. Various kits, testing kits are available. Even pregnancy kits are available. Various type of. Though, if this way we are able to identify various diseases in time, prior in time, the patient will be can be given a very fruitful treatment. And he will be deprived of all the harmful effects in this regard. So I think we are very much acknowledged by your esteemed knowledge in this regard, and hopefully we would have been able to understand a particular perception in this regard, having to understand the sensory, the micro nano sensor based perception of Ayurvedic diagnosis. And I request the esteemed audience, if they have any queries in this regard, they can please put their questions in this regard. I invite the audience. if they have any queries in this regard. Some of the audiences have sent me the WhatsApp messages. They was requiring your mobile number. I will share with them because they would like to have the interactions with you at all, because they must be very much interested in working in this field. So I would share your number with them also, and that would be naturally very much helpful for the for them as well for the Ayurveda also.
Sure,
Sure, sir. Any queries? If anybody is having, he can raise his questions here.
So, as an initiative, what I can say that to understand or to help the diagnosis in the Ayurveda, what we are doing is one is a smell based, so body smell or breath analysis. That is one area which we have identified, and we will be pinpointing some of the requirements, and some of my PhD students are also now interested in this area, and they will be working very closely for a gas sensor for diagnosing some smells. We'll be also looking for some taste related analysis using some of these sensors, which will be useful. So we are developing those requirements specifically with your team, Dr. Rakesh, that side.
Yes.
So you can pinpoint what are the requirements, and so that, because my students are electrical electronics background students, they do not understand that part. We discuss together, probably we'll be able to solve some of these issues.
Naturally, we have been there together in earlier meetings in this regard in our project, and we have been discussing all these issues regarding, as you said, you are specifically focusing upon, because it is your requirement. We can share whatever you need to be developed accordingly, as per your technology, and we can share our experiences. We can share our whatever the knowledge available in our decks and whatever the experiences we are having, and on that basis, we hope that we would be able to develop something fruitful in this regard regarding developing the micronuances in terms of smell or taste or gas, as you were discussing. So sure, as in the last meeting also we discussed that we will be there together, and your students, and we will be forming a group, a students group, that they will work together and find out something special, something good in this regard. We will be working in this field, sir. Sure, sir. Any further question, query? So okay. One more thing, if we are not having right now, we can have a discussion later on also, and you can share me as well. I will share the number of Professor Ajay Agarwal ji, and you would be having any query, we can meet it up in the later stage also. So I think
Rakesh, you may share my email ID also. So if they want to write, they can write.
Sure. I will share your email ID in my groups. Either if you are having, you can declare it right now, sir, on this in this channel. You can speak it out, and we will be having this lecture online as available as well on YouTube. They would be able to note down.
I'm sending my email ID also.
Email ID and mobile number as well. So it could be recorded in the YouTube channel. Yes, sir.
My mobile number also and email ID.
Yes,
so Ajay Agarwal at iitj.ac.in.
Yeah.
And mobile number is 8058598903.
Sure. And I will as and when needed, I will share it onwards also, and as per the requirements also. So with this, I think we come to the concluding session, concluding the moment of this lecture. So, I would like to acknowledge the dignitaries present in this program. First of all, I would like to thank our Honorable Vice Chancellor, an esteemed visionary personality who always is engaged in the development of Ayurveda and this Ayurvedic project, which is being conducted in collaboration with IIT, is also his vision, and we are working in this field onwards with great zeal. All our experts are working with great zeal. We are having regular meetings in this regard, whether in IIT or whether in university campus. So I really, from the bottom of my heart, I acknowledge my great gratitude to our Honorable Vice Chancellor, sir, that he is always motivating us and guiding us to move forward and take new initiatives in this regard. Thank you, sir. And I would like to thank Professor Shantanu Chaudhary ji. Though he was not there in the lecture, he has been busy in some schedule, but he has been always a guiding force, and whenever we happen to meet them, he always inspires us to do something innovative. Moreover, I acknowledge my gratitude to Professor Mahendra Sharma from this university, Professor Dr. A. Nilima, Professor Dr. Pramod Mishra, Professor Chandan Singh ji, Professor Rajesh Gupta ji, Dr. Manisha Mittal, Dr. Rajaram Agarwal ji, Dr. Manoj Adlakha, Dr. Prem Prakash ji Vyas, Dr. Ritu Kapoor, Dr. Meeta Chala, all and many more were present in this lecture from university. Dr. Sumit Kalra from IIT and Dr. Gaurav Soni. This is what I just want to share, that the dignitaries from across India were present in the program. Dr. Gaurav Soni from Shillong, Dr. Hemant Toshikhane from Vadodara, Dr. Kavita Kond from Maharashtra, Dr. Nidhi Srivastav and Ravi Srivastav from Jabalpur, Professor Prahlad Rakhu from Kasganj, Agra, Dr. Anurag Singh Chandel, UP, Dr. S.S. Chopade from Tilak Ayurveda Mahavidyalaya, Pune, Professor Vijay Lakshmi Gautam at IMS BHU, Dr. Sudesh Kumar Bhabu, Dr. Sachin Sharma, Professor Om Prakash Dave. He has been our faculty. Now he is retired from this university and is working in Paru University, Vadodara. Professor Om Prakash Dave, sir. Professor Pranab Jyoti Vaishya from Assam, sir, Guwahati, Assam. People who were far from across from India are present there. Dr. Swati Garg, Bhopal. Dr. Tikendrajit Sharma, Assam, Guwahati. Dr. Avinash Srivastava, Lucknow, and Dr. Dilip Kumar Goswami, Guwahati, Assam. So many more would have been also there. Maybe possible I am not identifying them. Some of them are having some showing their connection in others' names, so that may be possible that I may not be able to identify them. But everybody of the faculties who have been here, I really pay my gratitude that they have given this valuable time and to attend this lecture, and our beloved PG scholars from across India and this university and from IIT as well. They have been constantly present in this lecture, and no doubt they would have been regularly learning through this series something new, something innovative, and they must be in their career following all these initiatives and to do something at their own. Moreover, our technical staff from IIT, from IT section university, Dr. Hari Singhal and other Mahendra Sankalaji and other Manjeet Singh, the other technical staff, they have given this technical support with wholehearted support. I thank all of them, and especially Professor Ajay Agarwal ji. You have given us your valuable time. You must have prepared all this perception, because since beginning when we contacted you in this regard, there were some issues that how we would be able, because you belong to something different stream, and to persuade with the knowledge with the Ayurveda fraternity, naturally it would have been a little bit a concern of your lecture. So you have taken it very seriously, and I think you have gone with the details in a very easy manner, and so that we can understand all whatever you have mentioned in your lecture. And this lecture has naturally been very much knowledgeful. I personally very much acknowledged by your this esteemed gesture. Thank you once again, sir. And hopefully we would be there whenever we would be having you in this regard. If you would be having something new to share with us, we would be there. And this platform, Center for Human Resource Development, Ayurveda University Jodhpur, is always there, and we are here for the orientation of, or the extension of the knowledge to our fraternity across India, across nation. Thank you each and everybody who have been, and whosoever I have left, I couldn't name them. Everybody who has been present here, I please accept my wholehearted gratitude to be present in this lecture, and thank you once again. And I think now with this I conclude this session. Any queries, any issues, you can contact Professor Ajay Agarwal ji. You can contact me, and we will share your views to Ajay Agarwal sir, and hope that we would be able to hope we can manage at least something to solve your queries. Thanks a lot. With this, I think we should conclude now with this session.
Thank you.
Okay, sir. Okay, sir. Thank you. Thank you very much.